Hello?
Hi. I'm just
Hi.
explaining to Jesse that I'm having trouble with my Linux.
Oh, yeah. So just having trouble like getting it to run or install or what?
Yeah, run install, I don't know. It's sitting there looking at me and I can't get it to work.
Oh. What build is it? Like what kind of a Linux?
Well, I've got I downloaded the latest Ubuntu, and then I've got Windows subsystem for Linux.
Oh. I don't know. I heard that Ubuntu might be having problems with the last build, but I don't know. It's pretty platform or it's pretty platform independent sometimes.
Well, whenever I try to, when I click enter, like I type in what I'm supposed to go find the Windows shell, it disappears. So I'm not sure what I'm supposed to do about that.
Yeah.
It disappears. Press enter, the thing will shut off.
Yeah. No, it's a shame. I don't know. I don't know much about that beyond like what I just said. So
Well, trying to get the tech at the university to help me, and he says, "Well, why do you want to do that?" And I'm going, "Because I want to download Morpho."
oh yeah. Of course, everyone
Why?
does.
Because I'm getting kind of fed up with my console multiphysics.
Yeah. Well, that's all the rage, Morpho. Yeah.
Oh, is it?
I don't know.
I don't know. But let's try a command line program rather than a graphics one. I don't know.
Yeah.
The graphics one just wants to give me springs to work with, and those are not linear, so I can't really use them, but I'm not sure. I'm not sure what to do.
So it's like Jesse's here. It's a robot. Yeah. So yes. So we had a number of people apply for uh, Graph Neural Networks project for GSOC, and we had I think 5 applicants. That's pretty good. So I'll be going through those with some of the mentors. I know that Mayuk and Jiahang from last year. You remember Mayuk, of course, is involved in DevoLearn, so they're going to be mentors this summer for that project as well, in some capacity. So we'll be going through the applications on that, and then we'll be making a decision May 4th. So May 4th, we'll have an official announcement, and hopefully we can get more than one spot. I don't know what the constraints are on that this year, so it should be. Hopefully, we should have a fun summer, be able to do some things. I also got a message from Hari Krishna in the Slack. He's from last year, Hari Krishna Pillai, and he was asking about axolotl data or data from one of the specialty microscopes that we have for.
Well, it's May. Maybe I should go introduce some males to some females and see what happens.
Yeah.
Oh, they've got my Wi-Fi working up with the other computer.
Hello?
Oh, hello.
Am I audible? Hi, Bradley.
Hi, how are you?
I just want to give my. I told you like I was going for a village visit, and I'm staying in a university actually here. Some kind of what we call. Just let me show you. There's some practice going on here.
Yeah.
I'm kind of middle of that. I couldn't attend to this week meetings, but I will give my small update and I will leave. Is it okay?
Yeah. Go ahead.
I worked on the blog section actually. It has so many bugs. I was trying to solve them, and I was working on Hugging Face model to host the DevoLearn complete part. Maybe I will do it by this weekend. I am going back to my university by day after tomorrow. Then I can work. Then I can work. Start. Can I? Then I can work properly. And I have implemented the instant segmentation model also. I wrote some code. I'll share the link with you, the whole code part. Maybe you can review it. I will give access to you.
All right.
You can review it and let me tell what I, and I'm writing the code with commented code. What are the results will be there, how the code works and everything. These are my updates. And the blog was it is really tricky parts. I'm trying since 2 weeks to implement it, but so many errors are coming. I was using Hugo framework for that. It was creating some problems. Maybe I will solve it by next weekend, and I will let you show you on next weekend.
All right. Yeah, that
If
sounds good.
if any repositories are need to be created, I will mention you. Please create them. You need to put the whole Mayuk and Minux blocks in that.
Yeah.
So that's the problem.
Okay. Yeah.
I'm really sorry for this interruption in the meeting. I just want to let you know my work. I'm really sorry.
That's okay.
Okay.
Yeah.
Anyways, thanks. Yeah.
It's a party. It sounds wonderful.
Yeah.
Yeah. Anyways, bye. Have a nice weekend.
Okay.
Bye-bye.
Yeah. You too. Bye. That was Sushmanth. Yeah, we were talking about doing some. Well, he wants to, work on putting blog posts together. He wants to do this not just for GSOC, but for like a longer term project. So, like for his schoolwork. So, yeah, I mean, we had a number of people. Well, we had some work on the website. So we have this website on Weebly.com, and that has a lot of the core stuff for the entire group. And then we have another website on GitHub.io, which is sort of a mix of things, special things that we have. And I'll show this. I'll go through the screen share here. It's a mix of things that we have up here. So this is DevoWorm AI. It's actually DevoWorm.github.io, and we have things like what we call the DevoZoo, and we have like an onboarding guide there, as well as an onboarding guide on our GitHub. But one of the things we have is this DevoWorm AI, and this is something that Sushmanth has been working on with a few people. So this week we had a pull request from Jiahang Reddy. So Jiahang worked on this 2 weeks ago, issued a number of pull requests, and then made blogs, which is what's happened today. And so this is Sushmanth emerged that commit yesterday. So congratulations to Sushmanth and Jiahang for doing that work. But more generally, I wanted to bring attention to the DevoWorm AI, which is a sort of a repository of some of our models. So we've talked about a number of models over the last few years, and this is kind of a place where we can put some of these models. We have segmentation tools, C. elegans models. So the DevoLearn is that's on Hugging Face. That's what Sushmanth was talking about. That's where we have some of our segmentation models broken down into different types. So we have, let's see if I can find these. This is going to send me back to DevoWorm. Okay. The organization. So we have a number of different models on Hugging Face. We have the lineage population model, the nucleus segmenter model, and the membrane segmenter. And so those are on Hugging Face. And then we have DevoWorm AI, which is inclusive of things like digital bacillarea, general segmentation tool, C. elegans model, and so forth. So there's a lot of stuff up here that might be of interest to people. There's Jiahang. Well, Jiahang. So we still need to get a lot of that like better organized. I mean, it's always a constant struggle to organize things in a way that's really useful to people. But those are our resources for a lot of the Hello, Jiahang. How are you? That's good. So if you want to, I don't know if you want to say anything. If you don't, it's okay. You can put it in the chat. Anyways, I was just talking about our sort of where we put a lot of our models and our AI models. So we still haven't really updated it. It made a push to update it since last year. So we don't have any of the graph neural network stuff up. We have, we have a Basileria stub, but that may need to be updated. Certainly some of this needs to be updated. I'm not really sure how we should do this. I'm not sure if they hit Geothi and Sushmanth have been updating things, but I'm not really sure where that's translating onto these pages. So I think this one was updated and this one, but I would really like to go through it and make sure that we have, even if it's just like going through and making a video of like what we have for people so they can see what kind of resources we have. Because it looks like, you go through there and you don't really know. But anyways, that's something to think about as we head into GSOC, because that's something we'll be doing a lot of documentation. We'll be doing a lot of updating things, and then of course towards the end we'll be putting together an exposition of things for the projects, but also to sell some of what we've done. And we're not doing enough of that, I don't think. So, any questions? Comments?
I need to look at some of the online stuff more. I haven't been doing that.
Yeah, well, I mean, it's not. I don't know how useful it'll be for everyone, but just for like if you're into machine learning, the machine learning part of it, that's definitely a focus. At one point, we were looking at doing DevoZoo as like and it still is. It's just kind of we haven't really done much with it. It's for model organisms and getting secondary data for model organisms. So we have data for things like spiders and Drosophila and C. elegans, of course, and Basileria, just to have all those things in one place.
Anything about the axolotls? Or?
I don't think we have any. Well, we don't have any data for the axolotls, but I'd like to update it a little bit more too, because we, it was like the idea was like we would take like a model organism, have some data for people to work with, maybe some information about the organism, and then that would be like a place you could go if you wanted to do some comparative work, ideally. So you could grab like 2 or 3 species and try things. But I don't, we haven't really spent a lot of time on it in the recent past, so it's not.
Okay, well, I've been doing modeling instead of actual getting my hands wet.
Well, yeah, I mean, ideally too, it would be like if you wanted to model something in an organism, you need data or models, because that's kind of what we want to do. We want to encourage people to use models across species or models for a certain species, because not every species is going to use the same type of model. So like, some organisms, you might have a connectome or a tensegrity model or whatever, but they're probably specialized for that system. So,
well, one could come up with a sphere that is changing inside. Yeah, that's one thing I thought of doing for amphibian embryos, because they're producing basically salt water and pumping it into their interior, and then they become stiffer. Like,
Yeah.
that's called the blastocele, and I swear it's waste material that they pump into the middle of the blastocele, but I'm not sure. It makes sense. It's kind of waste material, but
Yeah.
anyway, it's a fluid, and it's I don't know how many people have done testing on what salts are in that fluid.
Yeah.
I'm not a biologist.
Yeah, I mean that's the thing too. Like a lot of the computational stuff is, people have an uneven background in biology, and then, especially if you're a biologist, you have a pretty uneven background in modeling and computation. So it's always like one of the things we want to do is to try to even that out somewhat, which is tough. I mean, it's hard to, like, biology has so many little different sub areas that
Yeah.
yeah.
Well, I've figured out that I'm, of course, more of an embryologist, but there's embryologist, homeostasis, and then cancer biology. There seem to be three of them, and so since I know more about the developmental biology, I tend to read more about that because I understand it better rather than the cancer biology. But it's all related.
Yeah. So you said homeostasis biology, or what was that?
Yes. I don't know what you call it, but that's what doctors work with. It's supposed to be this way.
Oh, right.
But
Yeah.
then it goes off into cancer, and then sometimes you get development happening as well. So
Yeah, that's that's kind of yeah. I mean, I guess there's the anatomy, and then there's sort of the regulation, and then maybe like
physiology.
Yeah, physiology, I guess.
Anatomy, physiology.
A lot of cancer biology is like looking at gene expression profiles and then looking at a cell and saying, that's a cancer cell, or
Yeah, and if it's got a different elasticity, it's often a cancer cell.
yeah.
If it's different from its surroundings, and quite often, they can be stiffer or looser than the surrounding tissue, but more often it's stiffer, like they form a lump. But that's not always the case. Anyways, the stuff that I've been reading.
Yeah.
Mechanics of biology.
Yeah. Well, I think that's an interesting kind of conversation to have. Is this, where do things fit into different areas? Where do the fields begin and end? Yeah, but I mean, so yeah, you're definitely doing this stuff with tensegrity, and I think that's a great direction. It's just a lot of biology is really gene centric. A lot of the stuff I've seen, anyways, and there's stuff, of course, that's with physics.
It's becoming. This is the physics of it, and it affects the genes this way.
Yeah.
And they've got the full spectrum out there now.
Yeah, I think though that a lot of the physics oriented people are in like biophysics departments or in physics, and it's like there's definitely a split between like people who would assay a bunch of genes or get like next gen sequencing data set and like mine it, versus people who would look look at the physics intently, like the mechanisms. And it's just a
they're
difference in the.
they're looking at how the nucleus changes shape and how that affects gene expression. So there's a lot of that going on. There's, yeah, at least I've run across papers about that. Of course, I'm more interested in how the cell changes shape, and then maybe it changes the nuclear shape, but
Yeah.
so I'm still on the other end of it.
Yeah, I think I'm gonna move on to some other features that I have here. So last week I had a scheduling conflict. So I'm sorry if I missed the. Apparently, a lot of people, I didn't send out a message. A lot of people didn't know what was going on. So I apologize for not making it clear. I mean, we had mentioned it the week before, but and so I want to go over some things now. So we had I found a couple things this week that were interesting that were kind of following up on some of the work that we've talked about in previous weeks. The first is this article, New Models Shed Light on Life's Origin. And this is a press release on this bit of work I think it's a paper. The research reveals clues about the physical and chemical characteristics of Earth when life is thought to have emerged. And then, this is a area of early life research where people were doing things like looking at astrobiology and other things to get clues on how life emerged and started to evolve. So this is the summary of this is researchers from the University of Rochester and the University of Colorado Boulder used experiments in zircon chemistry to build more accurate computer models of fluids that act as pathways from inner Earth to Earth's surface. So this is sort of Earth science meets biological science. The models allow researchers to simulate what metals may have been transported to Earth's surface when life first emerged about 4 billion years ago. This research has important implications not only for discovering the origins of life, but also in the search for life on other planets. So this is, we talk about events in Earth history, like the Great Oxygenation events, which was an event that increased the amount of oxygen in the atmosphere, and it enabled life to sort of hit a new plateau. So, it allowed for new things to evolve, new capacities of life to evolve, and so forth. In this case, you're talking about sort of the milieu of early life. So early life was not necessarily in a cell. It was maybe in a vesicle, which is like a little cavity that has metabolism, basically. It has chemicals in it, and it has to synthesize this self-sustaining synthesized set of reactions. We've talked about this in terms of the hypercycle. So, this is the kind of thing that people are interested in. And so there are a couple ways you can do this. You can either look at other planets and say those places are good candidates for life. Let's see what, that the planetary history is there, the planetary makeup. You can look at Earth and say, how did life first evolve on Earth? Think about early Earth, maybe back in the Proterozoic, and, what was the Earth like then? And in this case, what they're doing is they're saying that certain metals and elements have to be available at the time that life is synthesizing in order to sort of give it a kickstart or give it the raw materials to start. So the first signs of life emerged on Earth in the form of microbes about 4 billion years ago. While scientists are still determining exactly when and how these microbes appeared, it's clear that the emergence of life is intricately intertwined with the chemical and physical characteristics of early Earth. And so they interview a couple people here. They talk about how life could have started differently or not at all if the early chemical characteristics of our planet were different. So, a lot of people observing Mars note that Mars may have had a similar early history to Earth, and even Venus maybe had a similar early history. But at some point that diverged, and so right now we don't think that there's any life on Mars. There may be some microbes, and Venus certainly not. Maybe in the upper atmosphere of Venus, but certainly not on the surface. And so, why is that? Why are these three planets are so different now? And so that early history kind of shapes what happens later. And so that serves as sort of the basis for it. And so this is what they're kind of interested in. This model, allows us to sort of model that early milieu. And so metals are important in the emergence of life. And so when hypotheses are proposed for different origin of life scenarios, scientists have generally assumed all metals were available because there weren't studies that provided geologically robust constraints on metal concentrations of fluids from the earliest times of Earth history. So, we didn't really have, this is a good example of sort of how to build a model and what to include and what not to include. You can include things as sort of constants, meaning you just assume that they're in the background, that they're present at a certain rate, or you can exclude things you don't find important. So you could build a model of early life that didn't have any reference to sort of the metallic contributions. You can build a model of early life that assumes that the metallic contributions are constant. But you can also build a model where you are actually looking at the variability, where there are, parameter values that fluctuate. And so that changes the outcome of your model. And so you can find some really interesting results when you simply just include things in your model that you didn't think were important before. So this is a shortcoming of a lot of models. To address this shortcoming, Trail and McCollum studied the composition and characteristics of fluids in the lithosphere, or the outer layer of Earth that includes the crust and upper mantle. So this is actually back when the surface of the Earth was forming out of molten lava. This is the lithosphere, and it's cooling but not entirely cooled. So this is where you're getting really early. This is really early Earth, and these lithospheric fluids are key pathways to transport dissolved parts of rocks and minerals between Earth's interior and hydrothermal pools in the exterior where microbial life could have formed. So this is where you're getting a lot of transport of things to the surface and the formation of bedrock and things like that. You start to get hydrothermal pools at the surface, and this is where microbial life could have first started. And so a lot of things are being transported around, as actually we see all the time now. We have volcanic eruptions, we have erosion, we have drawdown of different chemicals from the atmosphere, and so forth. And so, this isn't like totally irrelevant to modern Earth. It's just that this is particularly relevant to early Earth. So they can't really directly measure the metal fluxes and where they were present or absent billions of years ago. But you can infer what metals and what concentrations they're at for your model. So you can kind of figure, again, add this in as a parameter, fluctuate the value, and see what it does to the simulation. And so we can get this from geologic information from billion-year-old rocks and minerals. We can do experiments in high pressure and high temperature. We can apply this in different ways. And so, this helps us understand this. There's a paper associated with this, Trail and McCollum, relatively oxidized fluids fed Earth's earliest hydrothermal systems. And that's a recent issue of Science. So this is the paper here. And so, So they're they're looking at the chemistry of aqueous systems. All right, so that's...
What is it called again? Oh, there it is.
Yeah, this one here. Relatively relatively oxidized fluids fed Earth's earliest hydrothermal systems.
Oh, okay. Yeah. Not something I'm studying, but
Yeah.
it sounds interesting.
Yeah. All right. And then the second feature is, again, on sort of this extraterrestrial life theme, but this is from a different perspective. So this is actually life evolves. Can attempts to create artificial life evolve too? So do efforts to create life by cooking up imitations in computers, robots, and molecules point towards a universal definition of biology. So this is a recent article here from Scientific American. So the question here is, what is life? We're trying to figure out what life is, and so we can get down to these building blocks that were around right at the earliest points in Earth's history. Where we know life was synthesizing. We just talked about that. So today our knowledge is so advanced that we can precisely manipulate life's building blocks, the DNA, RNA, and proteins, to build biological machines and engineer new genomes. Yet despite all we know, no universal consensus currently exists on life's fundamental definition. And so we know that life exists on Earth, but this is an N of 1. So we know maybe that life probably exists on other planets, but we don't know anything about it really. So we still have this N of 1 problem. And that is, we only know of one way that life emerged, and that was the way it emerged on Earth. And so if it appeared on other planets, we don't know anything about that. There's also this idea of panspermia, which is that life came from outer space, from different other planets perhaps, and transported via meteorite. But again, we don't really know anything beyond what we see on Earth. So this is an N of 1 problem. And so we can make a lot of predictions. We can build models like we did with the early life paper we just finished in the group. But at the end of the day, we don't know anything else about how life might have evolved. So in A life, however, people have been working on this for a long time. People have tried to build models of life-like things and sort of synthesize them from sort of a standstill or sort of an initial condition. And so in A life, we've done a lot of this. It's the systematic attempt to spell out life's fundamental principles, either by studying lifeless natural systems that exhibit lifelike behavior, or by building artificial systems to compare against nature's creations. Many of these practitioners, so-called A-lifers, think that that's the best way to really understand what life is. Build first and explain later. That's what they're proposing as a way to get at this problem. So you build a model. It's a sort of a generative model. You run it, and then you see what the results are, and you try to analyze those. You don't assume that certain results will be most probable from the get-go. You just build the model and let it run. So really, no one's been really successful at making actual life, but that's what you're doing. You're doing, you're taking on a pretty big target. So whatever you do, it's, I don't know if it's a dismal track record. That's what they say in the article. Takashi Ikegami, a complexity scientist at the University of Tokyo, is tired of such complaints. His field is just like any other basic science and seeks knowledge for knowledge's sake. So asking about the point of A life might well be missing the point. The existence of a living system is not about the utility of anything. Some people ask me, so what's the merit of artificial life? Do you ever think, what is the merit of your grandmother? What is the merit of your dog? So basically, it's not really easy to say what the merit of studying life is, other than to study life. And it's a pretty big question, so there are a lot of ways you could do that. So this sort of field of artificial life, You can study it. You can look at like the origins of life, which is nice. It's a sort of a trendy or interesting topic, but there are a lot of other things you can do. You can create novelty generators or systems with endless capacity for creating novelty. You can do other kinds of things like look at more focused problems in evolution. So this is something that, can stand in contrast to AI. So currently in AI, you can build these monstrous deep learning systems, but at some point, these systems can't learn anymore. And so what does it take for a system to continue to learn? Nobody knows. So this is where, we have this sort of idea that in AI, we have to train our models with data. And that's the only way it can really learn, but it's also the only way it can continue or sustain itself. So you train it with data, then you have, you ask it a question or you have a set of outputs, and that set of outputs is based on basically the training set and then the testing set and identifying things in the testing set or responding to a prompt that you give it. So in A-Life, things are more combinatorial. You can generate new possibilities based on sort of the structure of the system. For example, you can have a set of genes that mutate, and those mutating genes can produce things that you've not seen before. So it's really, truly better in the generative sense than a lot of AI models because it allows you to generate new combinations in a way that's not like, say, what you would find with a large language model where you're just combining things. We don't really understand how it combines things. A-Life models, we actually would understand how it recombines things. There are rules for understanding how, for example, genes with mutations, there's a mutation rate, for example. There are different operators like crossover we can apply, and then you can actually measure the output of the model at some point and compare it to the initial condition. So we have methods for understanding what the changes look like. And so, this is a nice article. It kind of goes through the history of the field of A-Life. It started in 1987 at the first A-Life conference. And, kind of goes back to the middle of the 20th century where people were trying to understand how machines can think and replicate. And yes, actually John von Neumann and Stanislaw Ulam, they worked on these kind of problems together, building things like the von Neumann machine and other types of, or the von Neumann replicator and other types of things. And of course, this evolved into the modern A-Life systems that we have now. So this is animation of the progression of a breeder, which is highlighted in red. Breeders are a class of cellular automata. So this is on a cellular automata grid. This is where you get different behaviors from a single set of initial conditions. They kind of evolve across this grid. A lot of Conway's game of life involves movement and evolving movement in different directions and different types of behaviors, and then characterizing the behaviors in some way. So this is an example of Conway's game of life. This came in around 1970, and then we've been working on things such as soft A-Life, which is software like this that's generative that can produce things that look lifelike, and then hard A-Life, which is the creation of robots and other types of hardware. And then there's this new branch called wet A-Life, which concerns the creation of synthetic organisms using biochemistry. So what we were talking about just now is sort of a mix of soft A-Life and wet A-Life, where you're using software simulations to build some sort of biochemical system. So this is a nice article. Kind of goes through a lot of the history of the field, and it goes through some of the applications. And I don't know if there's anything more to say about it. I guess they just kind of give a critique of the field, and maybe that it's doomed to succeed, but I don't know if that's true. I mean, that's up to people who want to do things with it. I think. But you can test a lot. You can test a high evolutionary hypotheses using AMIF. You can do a lot of things. So you can't necessarily increase the N, but the N of observables, but you can increase the N of observables if you think that like a simulation is equivalent to what we see in the real world. But it's again, we have to have this caveat of simulations aren't really like the real world. We kind of control what's in the simulation, and thus that changes what we're observing in these simulations. So, it's a good article. It's from Scientific American. It's Life Evolves. Let's see. Life Evolves, can attempts to create artificial life evolve too. So let me grab the title, put it in the chat. All right, so it looks like Jiahang had to leave. He was just checking in, I guess. I'm glad to see you. Look forward to this summer. Hopefully we can get some good work done. So any questions, Susan or Jesse?
No, I'm just listening.
Yeah. So actually, the next thing I wanted to talk about was I wish Jiahang had stuck around a little bit longer because I was going to mention this, but so this is a Learning on Graphs conference is going to be back for a new year. We had Learning on Graphs in 22. This is something we thought about submitting to, but weren't able to submit to. But this was the inaugural conference, Learning on Graphs, and so this was a nice conference. It had a lot of. This is actually the video, that's the YouTube channel of their talks. But this is the Learning on Graphs for 23. So this is.
Can you see it on YouTube?
Yeah, this link here, log 2022 on YouTube. So this is logconference.org, and you can
put it in the chat.
Yeah.
Okay. I'm sort of doing graphs, right?
Well, yeah. I mean, this is graph neural networks more, but this is the topics range from sort of machine learning to some topics in mathematics. So last year was really interesting because they had, mathematicians, and they had computer scientists, and they had machine learning people, they had modelers. So I don't know what qualifies as a good submission, but the submission deadline's coming up, I think this summer sometime. So we'll have to target it. But the conference is going to be at the end of November, and it's it'll be a nice opportunity even if we don't get anything accepted from the group to do like to attend. Last year I did a blog post on it where I went over a number of different areas of interest that were out there. They laid out, I think, the field very nicely. They laid out what they're interested in. So these graph neural networks are neural networks that have some sort of geometric structure to them. So instead of being like just these layers or these hairballs or these circular networks, they have some structure. This is not a good example of what a graph neural network is. Basically, if it's any piece of data that you have that has some graph structure to it, and graph structure is loosely defined, people apply graphs to a host of problems. But basically, if you have a graph structure in your data, you should be able to extract it using a graph neural network, build some sort of representation of it, or embedding, and then do computations with it. So there's some really interesting domains, although the domains focused mostly on molecular biology and medicine, and then on other problem areas. So there isn't really a lot of what we're doing. Like embryos, there wasn't anything on embryos, there wasn't anything on, phenotypes, things like that. But there was a lot of stuff on like characterizing protein structures or molecular interactions. And so that was something that's like a basic standard systems biology problem where you want to build like a protein-protein interaction network, and you have, um, you know, you have a graph structure already there. So then you just need to apply this technique to it, and you get a better graph. Apparently.
Do you have what sort of program are they using for this? Is it like the Morpho I've run into and it's Linux based, or?
Oh, so yeah, so this is like very specialized. It's they're doing like machine learning algorithms. So they're writing their own algorithms and implementing them. Or sometimes they have, I don't think they have any specialized software packages for this. There's some Python toolboxes that you can use to build these, but they're in pretty early days. So that's the other impression I got, is that it's kind of like, in some ways, it's like a Wild West where they don't really have the tools built, so people are building the tools and seeing what works. There are like a host of issues, like how expressive is a certain algorithm. So like if you apply this algorithm that generates a graph from your data, how many ways can it express your problem? And some methods are not very good at expressivity, and some are very good. And so that means that can you really express the entire content of your problem using an algorithm? Sometimes it's very hard. Like, for example, if I just use like a matching algorithm for some problem that required a lot of deep spatial structure, that would not be very expressive, because I would just simply be finding like surface level detail of my model, and it wouldn't really match my model, so it wouldn't be very expressive. But if I used an algorithm that matched that sort of deep structure, it could be like a graph neural network and like some time series tool, that might be better, because you'd be able to capture more of your data using that, capture more of the complexity. So that's one of the things that they talked about, I think, is really kind of relevant to what we're doing, which is, to maybe, propose some sort of approach to, back up from what we've been doing with graph neural networks and say, how do we propose to build like networks, or how do we express the networks that are in developmental biology, like in embryos or in other types of systems like that? There are these networks that are in like sort of embedded in the data. So, we have these problems like tensegrity, these problems like interaction that are, unique, because it's not just interaction at one point in time, it's interaction over multiple points in time. Yeah. And then consider like what kinds of things, what kinds of algorithms maybe could express that, as opposed to just throwing something off the shelf to the problem. So, part of that is what you're seeing with some of the programs you're using, is that they're made for mechanical tensegrity instead of biological tensegrity. So there's a lot of the biological stuff that gets mushed in there.
Well, I just made it sort of like what a cell is. Like I tried to use the same structure. And I keep getting asked, well, why would you make it a hexagon, and why do you have it hooked up like that? It's just like, because that's the way a cell is hooked up to its neighbors.
Yeah.
And then I tried to call it, what was it, a cable net, and I get yelled at. So, okay, you don't want to call it the cable net. It is, but I don't have to call it that. I'm just going to say it's hooked up to its neighbors. Yeah. I give up. Yeah. And it's a stable tensegrity because it's hooked up to its neighbors.
Yeah. Yeah. So, I mean, as you can see, there's sort of an open problem there, in that you have this notion of like, how do we get what we want out of the model? But we have to build a model that's like basically able to provide an answer. So
Yeah,
if it can't
yeah.
provide the
Well,
answer, then yeah, we have a problem.
well, I need some quotes on this actually because a cell, I mean, a tissue is the cells in a tissue rely on their neighbors
Right.
for their structure. And if they're no longer connected to the tissue, they become an amoeboid cell and crawl away or become or undergo apoptosis or something. Like I need to quote that because that's kind of what's going on with this. Like
Right.
so basically, let's make an unstable tensoid, even make it stable by hooking it up to its neighbors.
Yeah.
So that's maybe a way of saying that.
Yeah. You kind of need
Because that's the way it is in reality. That I'm just trying to mimic reality, kind of.
you sort of need a background structure for it in order for it to start. It's kind of like what they do with in bioengineering. They give like something a scaffold, where like if you're growing like an organoid or some tissue, you have to put it on a scaffold, and then the scaffold has to dissolve, and you have this. But it's not, it doesn't always work. Sometimes you fail, and the cells don't grow along the scaffold in the right way. So they have to provide all these nutrients and things along the gradient, and eventually maybe you kind of get what you want.
And you're never quite sure whether it's the right cell type.
Yeah.
That's my argument with it.
Yeah.
Oh, you think you made a nose? Is it real nose tissue, or is it like whatever you started with?
Yeah.
What is it?
Yeah. But the scaffolding is important. There are other things that are important too, but if it doesn't have that scaffolding, it just falls apart. And it's not just the mechanical scaffolding; it's the sort of that environment, like that milieu of chemicals that are there.
Yeah, and you need, so they've got some gels that are pretty good, and they've embedded the right chemicals in them, and then sculpted them to the right shape so that the cells are actually um, going around corners to get the right shape, and some of them work fairly well.
Yeah.
And still my question. This organ or whatever you've made, is that the same type of cells that you're getting in a real one that somebody else, say it's a nose or an ear? Is your ear the same? Does it have the same cell types as a normal ear? And how can you tell?
Yeah.
So, but cartilage is pretty good because it's normally a cells that develop in low oxygen or from synovial fluid. So they're not relying on a blood supply per se. So they're pretty. If you want to make that type of structure, that should work. Is all I'm saying.
Right.
Because it's more easily manipulated, and it's often the part of you that goes wrong, say in your knee.
Yeah.
We should have some success with that, and I know they're always heading for the spinal column and spinal cord, trying to recreate that. So there's a lot of interest and research in that area.
Yeah.
I just downloaded something the other day, and they've got a gel that they're trying to use to try to regenerate spinal cords.
Oh yeah.
Yeah. Anyways.
Yeah. I just find that sort of work fascinating. That we're learning a lot about that. So let's see. So I had some papers to follow up on from the stuff we were talking about last. Well, we've been talking about curvatures and modeling and things like that. And so I have two other papers. So this is Morpho. This is the Morpho paper, actually. This was on the archive under condensed soft matter, their condensed soft matter repository. So this is the Morpho paper. I guess it's like the seed paper for the software or something. This is from Tufts University. It's a whole group of people from Tufts, and so this is where they talk about an emerging theme across many domains of science and engineering is materials that change shape often dramatically. Determining their structure involves solving a shape optimization problem where a given energy functional is minimized with respect to the shape of the domain. An auxiliary field is describing the structure. Such problems are very challenging to solve and therefore a lack of suitable simulation tools that are both readily accessible and general purpose. So this is where they introduce Morpho, and they demonstrate the versatility by showcasing three applications at different areas of soft matter physics: swelling hydrogels, complex fluids that form aspherical droplets, so they're oblong shaped or non-spherical, to soap films and membranes, and advise on broader uses. So you have these soap films and membranes, which are surfaces that have like bubbles, and they're bubbles that have these edges that are continuous. So you have this network of edges for the soap film. So soap bubbles.
And they're like cells.
Yeah, they look
cells
like cells.
do the same type of packing as the soap film.
Yeah.
Or bubbles.
Yeah.
It's the same sort of foam shape usually.
Yeah. So then swelling hydrogels are just things that swell as a sort of a surface. So it's like a tissue growing, I guess. So a lot of these soft materials, you can use them as analogs to the biology, but also they have that sort of the same properties as the biology, like in terms of stiffness and things like that. So this is basically a shape optimization. They're using an energy functional, which is physics, basically a physics tool to determine the energy function and then to minimize on that. You constrain the energy function and you can get these minima that allow you to solve the problem. So one option to do this is level set methods, where you represent the free boundary of the system as a contour or a level set of the scalar function defined in a higher dimensional space. So you can capture changes in topology and handle them easily, but you need to formulate the optimization problem, you need to use a lot of sophisticated techniques. So in Morpho, they use the explicit discretization of the problem domain and associated quantities. So from these, Morpho is able to evaluate the object function of interest, as well as its gradients in Hessian, which is a mathematical operator, with respect to the mesh and field degrees of freedom. A number of algorithms for constrained optimization are then available within the Morpho environment. And the approach is similar in spirit to the highly successful Surface Evolver software. So this is 33 and 34. And that is these two references, the Surface Evolver, which is from the 90s, and then the Surface Evolver and the Stability of Liquid Surfaces, also from the 90s. So you can see this has a history. And so this is what they were doing. I don't know if you can link to the paper. Looks like you may be able to. So this is the Surface Evolver. This is a computer program that minimizes the energy of a surface subject to constraints. The surface is represented as a simplical complex. So this is actually goes back to graph neural networks, because what they're doing in graph neural networks is they're creating these graph embeddings. And one thing it has it's analogous to are types of structures that topological data analysis would be interested in. So it's basically taking a network and breaking it down into these cyclical complexes. So you're finding shapes within the network, like triangles, diamonds, and circles, and other things. So this is actually something that has, it's kind of strangely related to graph neural networks. So the energy can include surface tension, gravity, and other forms. Constraints can be geometrical constraints on vertex positions or constraints in integrated quantities such as body volumes. The minimization is done by evolving the surface down the energy gradient. So you're basically using the energy gradient to find the shape or the surface properties. This paper describes the mathematical model used and the operations available to interactively modify the surface. So this is a good paper for the origins of this stuff. See if I can put it in the chat. I don't have it in the collection.
Yeah, what I've been told is that tensegrity is a minimum energy system.
Yeah.
So it kind of fits with that.
Yeah.
No, I just need to convince the professors that I should try this.
Yeah. It's hard to do, but yeah. I
mean, yeah.
So let's see.
I just paid $1,000 for console, and you want to quit? Yeah, I want to quit. I'm tired with it. Anyways.
Yeah. So there are two other papers. There's this let's see, unified phase diagram of reversible, irreversible jamming and yielding transitions in cyclically sheared soft sphere packings. So this is a paper on surfaces and phase transitions, which we know in development and in soft materials, we have these jamming phase transitions where you have a bunch of particles and they're moving through a space, and when you pack them tightly enough, they undergo, at some density, they undergo this jamming transition where they go from free-flowing to sort of jammed into place. So they stop moving, and this is a phase transition, so it's a quick transition from moving to immobile. And so this usually happens at some density of packing. So if they're flowing in a pipe, if they're a sparse packing of particles flowing through a pipe, they flow through fine. If there's a denser packing, they start to slow down, but they don't. It's not a linear thing. They slow down a little bit and then they stop moving at some critical density. So this is something that they're looking at here: reversible, irreversible jamming. We've kind of talked about that in embryos too, where you get this, motility of cells in the embryo. There are flows of cells. There are other types of migration, but then you have these jamming transitions where things stop moving based on their packing. And so these are both reversible and irreversible. In some phases of development, you get a reversible jamming phase transition where things start to move again. And so these are transitions in development that aren't necessarily well characterized, but people see them in a microscope. So the abstract here is self-organization and transitions from reversible to irreversible behavior of interacting particle assemblies driven by externally imposed stresses or deformation is of interest in comprehending diverse phenomena in soft matter and in embryos, but we'll just say soft matter for now. They have been investigated in a wide range of systems, such as colloidal suspensions, which is like like mud or some other type of liquid with solids in it, glasses and granular matter. In glass phase transitions in glass, the behavior of glasses is well known. So, that's something, again, is another model system. It's interesting. In different density and driving regimes, such behavior is related to yielding of amorphous solids, jamming, memory formation, etc. So this is memory, not like cognitive memory, but like shape memory and other types of like things that involve hysteresis. How these phenomena are related to each other has not, however, been much studied. In order to obtain a unified view of the different regimes of behavior and transitions between them, we investigate computationally the response of soft sphere assemblies to athermal cyclic shear deformation over a wide range of densities and amplitudes of shear formation, or deformation, shear deformation. Cyclic shear deformation includes transitions from reversible to irreversible behavior with unjammed and jammed soft sphere packings, well above the minimum isotropic jamming density, which is this parameter. The transition corresponds to yielding. In the vicinity of the jamming point, up to a higher density limit, we designate this parameter, J sub sic. An unjammed phase transition emerges between a localized absorbing phase and a diffuse irreversible phase. So you have this sort of heterogeneity that starts to emerge. And so, let's see. The emergence of the unjammed phase signals the shifting of the jamming point to higher densities as a result of annealing and opens a window where shear jamming becomes possible for frictionless packings. So this is where you get this friction as a parameter. You get frictionless packings, you get shear jamming that occurs as a result of some of these changes. Below J are two distinct localized states, termed point and loop reversible, are observed. We characterize in detail the different regimes and transitions between them. So they do this, basically they're characterizing the materials, they're looking at these different phases of jamming and unjamming, and then they're putting it into a phase diagram, which is, let's see if I can get to the phase diagram. So this is, this is not a stress strain curve. These are strain amplitudes. So plot it that way.
That's not a stress strain curve.
No, it's not. It's some sort of, I don't know what it is exactly. This is, oh, go ahead.
One that relates the shape of the cell to the packing and its fluidity. So if it's more stretched out, elongated, it's more fluid. And if it's more like that hexagon I was talking about earlier, then it's more stable and considered a solid.
Yeah.
So it's the hexagon is a more stable form of it. It's not as fluid. I don't know. And this is some of the same type of study.
Yeah.
So it won't be a stress strain curve because that's not what they're looking at.
Right.
Yeah.
I don't know what strain amplitude. Well, I guess it's just kind of like looking at strain over time and looking at the intensity of it, or.
Yeah, I'm not sure if they're doing cell shape here. Well, I haven't looked at this. So.
Right.
What are the units? That's what you're going to. What is it? Strain amplitude.
Yeah. So I think the key here is that there's a discontinuity for PE, which is, I don't know what that is, but this is
is it the circumference of the cell to its area, sort of some ratio? Is that their P?
Might be, yeah. Yeah. I'd have to go back up into this point to see. Oh, the potential energy, actually.
Oh, potential energy. Yeah. Oh, okay. Well, that's different then.
The point is that there's a discontinuity at a yielding strain, so you get this basically this phase transition, or and then you're observing these different phase transitions in the.
Yeah. It's neat. You get a phase transition with tissue because it's got a J curve, so it goes along a J curve when you're in a stress strain curve. So it has one elasticity and then it changes to another elasticity. So it creates a J curve because it's got a transition. There's a transition from one elasticity to another with the stress strain curve of living tissue. Yeah. I have collected a number of them. I have a PowerPoint with a collection of J curves.
Oh, wow. Okay.
Yeah.
So this is accumulated strain. Here they again show this discontinuity. Jump in diffusivity is a function of strain amplitude. So again, this diffusivity, how easy it is for the things to move around based on a function of a strain amplitude, however they define strain amplitude exactly. And you have these different phases. So this is B here, plot of the non-affine path length L and the percentage of new collisions C nu. So this is C nu, this is L, which clearly differentiate all the different phases and all densities. And so you have these different phases, absorbing phase, unjammed phase, and yielded phase. So the phases are actually colored in. So this is down here, this is A. The star is yielded. I think the star is up here. And unjammed is this blue triangle, which are down here. So that's absorbing and unjammed are down here. Yielded is up here. It gives you a sense of where it is in that phase diagram. Then this is another one where you see regions in the phase diagram. So this is where you have yielded. This is, let's see, A is, I don't really label them here, but you can see that there are these different phases along in this phase diagram here. So these different regions that represent different states of the system.
Maybe that's what I need to be making.
Something like this.
Diagram.
Yeah. They're nice to look at. You can like kind of have an index, but you have to sort of predict for all these different values. So you have to do like 2 parameter sweeps to set it up. You have to do this parameter and this one, and see at different values what the value, and then you can define these lines, these boundary lines, and just color them in. Yeah. I mean, those are, they're useful. I mean, they do give you a lot of information about sort of the transition points, but they're not precise. I mean, if you were to go and test the system for every value, you may or may not, this boundary may or may not be like the thing you come up with, but it basically tells you where in that space you get a certain phenomenon. So yeah, so this is more of the same. So this is basically, there are a lot of connections to surfaces, biological surfaces. In that study, they didn't look too much at the biology. They looked at these different soft materials. In this paper, they're talking about jamming on curved surfaces. So I think Susan mentioned Timothy Atherton maybe 2 weeks ago. Okay. So
yeah, he's into morpho.
Oh, okay, yeah. One of the morpho people. So this is so the abstract here reads colloidal and other granular media experience a transition to rigidity known as jamming if the fill fraction is increased beyond a critical value. So the fill fraction, I guess, is how much is in a space. The resulting jam structures are locally disordered, bear applied loads inhomogeneously, which means that there's a lot of, heterogeneity in terms of how they compare loads across the jam, possess the minimal number of contacts required for stability and elastic properties that scale differently with volume fraction of crystalline media. So this is, again, I guess this part here is, it possesses the minimal number of contacts for stability. They scale differently with volume fraction, meaning that the minimal number of contacts increases not linearly. with the size of the jam. I'm not really sure. But anyways, here the jamming transition is studied on a curved ellipsoid surface by computer simulation, where shape evolution leads to a reduction in area, crowding the particles and preventing further evolution of the surface. So the particles get jammed into a smaller area, and then they can't, this kind of constrains the surface. The arrested structures can be unjammed and the surface further evolved iteratively, eventually leading to a rigid metric jam state that is stable with respect to motion of the particles and some specified space of deformation of the manifold. So you get this jamming when you constrain the surface, and you get a more packed system, and then you can actually that's arresting the structure, and then you can unjam it, and then this allows you to have these sort of deformations as a result of unjamming. This is kind of like in a tissue. When you have tissue formation, you have cells that start to double and then double again, and you get more and more cells, and eventually you get these jam states, and then you get this some sort of folding or bending or deformation of the sheet, and then you get this. So you get morphogenesis as a result of this sort of need to move to a different functional state. So you get this jamming, and then you need to unjam it, so you get folding, and that allows it to unjam. At least that's how I'm understanding it. At least the metaphor to development. The structures obtained are compared with those obtained in flat space. It is found the jam states and curved geometries require fewer contacts per particle due to the nonlinearity of the surface constraints. In addition, structures composed of soft particles are compressed above the jamming point. It is observed that relatively well-ordered but geometrically frustrated monodispersed packings share many signatures of disordered biodispersed packings. So that's, I'm not sure I understand that very well, but they talk about jamming here in the intro. They talk about some of the powerful tools that have been developed to classify the nature of jam structures. For hard particles, Torquato and Stillinger proposed a taxonomy of jamming based on the space of feasible motions available to the constituent particles. So this is where they can move in different ways in different directions, and that sort of is the determinant of jamming. So a packing is locally jammed, the least stringent category. If no particles are able to move, all the others remain fixed. So this is where nothing can move, like a traffic jam. It is collectively jammed if no subset of particles is movable, with the remainder held in place. So I guess this is where each cell is holding the other in place. So if one cell were to move out of the way or be removed, you'd get this avalanche of cells that start moving in response to that. Then it is strictly jammed if no collective subset of the particles can be moved at the same time as a volume-conserving deformation of the container. So this is where you can't move the particles at all. And then as long as you conserve the volume, if you change the volume, like if it's a pathway or if it's like an egg, and you increase the size, then the cells can move out. If not, they're not. So these are their three different types of jamming, and they're kind of technical definitions, but they're kind of important to development if you think about it. The category to which a configuration belongs can be determined numerically by solving a linear program, for which efficient algorithms exist. So this is the basis for what they're doing in Morpho. In contrast to hard particle packings of particles of soft, finite range potentials can be compressed beyond the jamming point. So you can compress cells beyond the jamming point. It's just that they don't move from the jamming point beyond. So they can be, so hard packings sort of follow these three conditions, and these are also maybe applicable to soft materials, but soft materials can be constricted even more. So because they're soft, they can give and they can compress. So that's something to think about in terms of modeling soft materials versus harder materials. They also exhibit critical scaling laws as the density is increased above the jamming point. This is true for the contact number Z, as well as the bulk modulus B and the shear modulus G. And these are all like different aspects of shear and stress and strain. So, these are things that soft materials are going to have that hard materials are not. These scaling properties reveal the nonlinear nature of packings near the jamming point. So there's some nonlinearity there that has to be accounted for as well. In flat space, monodispersed packings in 2D, in 2 dimensions, are highly crystalline. Disordered monodispersed packings can be produced only under extreme circumstances. However, for packings in a curved 2D space, which is where you have curvature instead of just this flat surface, that's where there's packing or no packing, crystalline order is frustrated by the surface geometry, necessitating defects and inducing strain in the crystalline regions of the packing. The question arises then of how geometric frustration affects the mechanical properties at the jamming point. So this is geometric frustration, where you start to get this nonlinearity, you get this curved surface instead of the flat surface. The curving introduces these instabilities, and then that introduces nonlinearities. So it behaves differently when it folds or when it curves. And so this is what they're talking about in this paper, or about soft materials jamming on a curved surface. And so they kind of go through some of the math here, and they talk about, all the different aspects of this. They give a mathematical model for unjamming arrested packings. They give, here's a good example, a picture of what we're talking about. So this is A and this is B. This looks kind of like an embryo, but it's not, at least not in what they're modeling here. This is A, which is the arrested structure produced by the evolution of an ellipsoid surface at constant volume with the particles colored by the hexatic order parameter. So this is where we have this order parameter from 0 to 1. And I think we did, there was a paper recently we talked about with Drosophila a couple weeks back, where they had this very thing, where they had these, cells that were sort of in an unstable regime versus a stable regime, and they were flowing. And they showed this kind of pattern where you get like pattern formation resulting from this type of packing. So I don't remember all the details, but there's an analogy to that. I'll have to dig that back out and look at that in more detail. But this is not an embryo. This is just a physical, a soft material, or actually maybe even a hard material. B is...
They would be related.
Oh, yeah.
So, yeah.
B is the unjamming motion found for the structure using the linear program. So this is where this is the predicted unjamming motion. So A is where it's jammed, and there are different order parameter goes from 0 to 1, which is just basically the parameter we use to say whether something is ordered or not. There are instabilities in these regions where closer to 1, and then stabilities where it's closer to 0, I believe. And then, or it could be the other way around. In any case, then this B predicts the motion for each particle that they would have to take to unjam. So you can see that there's differences in the sort of the unpacking. It's kind of like a Jenga tower. What part do you need to pull to get the whole thing to fall? It's not obvious from the get-go. You can model it and say, okay, you remove this piece and the whole tower falls, because they're not evenly, they don't all contribute to order in the same way. Some of these are highly ordered. So if you remove a packing from here, or you remove a particle from this part of the packing, it doesn't have any effect. If you remove a particle from this blue part of the packing, it has a bigger effect. So then that's what, but they're all going to be moving in a certain way so that they become unjammed. So again, it's like a traffic jam. What's causing the traffic jam? If you remove that, you can unjam the traffic. If you don't, if you just remove cars at random, you can't necessarily unjam the traffic jam. So there's that inhomogeneity, there's that instability, but then you can also unpack it. You can move the cells in certain directions, and so that's what this is. And then that becomes important in embryos. Actually, they do this, like I said, this unjamming that occurs when you get folding or curving that occurs in different tissues. So this is something that is relevant to that as well. So they talk about arrested jammings or arrested packings. They ask whether they're truly jammed or not. And then a criterion for isotesticity, which is where you have this even amount of stability. And this goes through a lot of stuff. And then they get into soft particles. So that wasn't even really focusing on soft particles. There's a soft particle section where they actually have an arrested packing of 5 particles. So they actually have this atomic example where this looks really relevant to tensegrity, because you have this very small example here of five particle system where you can actually look at the packing in terms of its center of mass and how they're all related. So this is the linear programming finds this unjamming motion representing rotation about the ellipsoid, but this motion is not feasible within the nonlinear surface constraint. So these arrows show where the center of the center of mass has to move in order to unpack this. So you see this, this is the predicted motion, but it may or may not be feasible given the surface constraint. So they give a lot of interesting stuff in this paper. I think this is really useful for maybe like understanding what Morpho is doing, but moreover, I think it's really interesting from a theoretical standpoint, because it's really kind of getting at this idea of packings and why they're important and how they maybe relate to networks. Because we have networks here. We have a network here in this 5 packing, and then we have another network up in figure 1 where we have this thing that looks like an embryo. This is a network, and we can move the network and change the topology in a way that's maybe leading to some sort of change or phase transition.
Okay, thanks for going through that.
Yeah, no problem.
I have to contact the author, Timothy.
Yeah.
Today and say I'm still very interested in this program. Having some trouble downloading it. Anyway, yeah.
All right. Well, thanks for attending the meeting. Again, next week we'll be back. If you have any questions, I'll send you those papers, Susan. I know you're not in our...
Well, Kathy was interested. He says that paper in chat or Slack ?
Oh, it says, okay, yeah, I can.
One.
I can put these in the OpenWorm Slack for Jesse. So yeah, check that, or I might put them in the other. We have another Slack, which we can use for that. So I'll get them to Jesse as well. Okay.
Yeah. Well, it's sort of very related to what I'm trying to do. So.
Yeah.
Thank you.
All right. Thank you. Have a good week.
All right. Bye.